Hello one and all. So today we are going to begin with PGT entrance examination of biology concept, where in the first chapter we are going to deal with biological sciences, where we just have 3 concepts to deal with in this chapter: importance of human welfare, branches of biology and biologists. Wherein this is completely memory oriented chapter. I have given nice tabulated forms which eases our preparation, and where we can memorize each and everything in a short term. Okay. So let's begin with the introduction of this chapter. Biology plays a crucial role in improving human health, agriculture and environment and industry, have based on the microbial usage in all these aspects, okay, where we are using microbes for all these purposes, be it in human health, agriculture, environment and industry. It helps in sustainable development, disease control, food production and biotechnological innovations as well, where we are utilizing all the techniques of biotechnology on microbes. And then microorganisms or microbes, though invisible to the naked eye, are powerful biological tools used for the betterment of the human life. These microbes organisms include like bacteria, fungi, protozoans, algae and viruses. Let's discuss about this unicellular organisms in detail. Like viruses, we will call them just as living entities or the living crystals, because it just has the proteinaceous coat and inside the genetic material. There is no protoplasm in that. But viruses are also, few will be beneficial and few are harmful. As such, we have bacteria. Bacterial usage is very broad, where we have broad spectrum of harmful bacteria and useful bacteria, where we have to remember the examples for sure. And we have unicellular fungi like Saccharomyces cerevisiae yeast, where we will be using that in baking industries, brewing industries. And then protozoans, which are animal like protists, where we have n number of types of protozoans like amoeboid protozoans, flagellated, ciliated and sporozoans. Best uses of that, and algae when it comes into. We have most of the unicellular algae under the green algae, okay. So many of them are used as food, some as hydrocolloids, some as food sources, some as single cellular proteins, okay. So all that we are going to look into now with examples. So in numerous sectors they are used like agriculture, industry, medicine, environment. Their beneficial roles far outweigh their pathogenic effects, making them indispensable to human welfare, okay. They serve as natural recyclers of the environment. Yeah, they serve as natural recyclers in the environment. They produce food, beverages, antibiotics and enzymes. Key players in sustainable agriculture through biofertilizers and biopesticides, and they also act as agents in waste management and bioenergy production, okay. Then first let us see all the microbes that are useful for household products. Microbes actively involved in everyday food preparation and processing at household level. First, traditional Indian fermented foods are made using natural microbial cultures. The microbes will improve food digestibility, they will add flavor and they will add texture, which increases or enhances the nutritional quality of the food. So first, if we see lactobacillus, which is the well noted example for us, which is used for producing curd and yogurt. The main action for this ferments lactose to lactic acid. Best examples of this enhances digestion and gut health. Next is Saccharomyces cerevisiae. As I said that this is used in baking, brewing industries. Best products by using the Saccharomyces cerevisiae or yeast include bread, idli, dosa batter that we make in household products, where this produces carbon dioxide for leaving, that is a puff will be formed, right, that is leaving. This produces carbon dioxide by conducting what type of fermentation? Alcoholic fermentation, wherein carbon dioxide will be evolved. Alcoholic fermentation 2. Next, leuconostoc or streptococcus. These are bacteria wherein they are used to produce fermented foods, where this adds flavor, texture, enhancement, and this is best example is to prepare pickles and cheese, that adds that pungent flavor for the pickles and yeast. So that is the bacterial examples for that are Leuconostoc and Streptococcus. Next, microbes in industrial products. So in industrial products, industry will rely on the microbes only to produce some commercial products such as organic acids, alcohols, enzymes and antibiotics. Let us look into different types of enzymes for each of the purpose. Okay. Microorganisms and cultivation in large bioreactors under controlled conditions. First, Saccharomyces cerevisiae, most of the examples will be repeating, like few of them will be repeating. For ethanol and alcohol, where this industrial user is alcoholic beverages and hand sanitizers. Next, Aspergillus niger. Immediately when Aspergillus niger comes, citric acid comes into our mind. So this is the product that it is producing is citric acid, which is used as a preservative, or this is also used in the soft drinks. Next, Acetobacter aceti. The name itself is saying the product of it is acetic acid. This is also said as commonly vinegar, where we will use this in pickling and as food preservative. Next is Lactobacillus species, which produces lactic acid, and lactic acid has enormous use in pharmaceuticals and as food preservation. Clear, right? So these are the industrial production of alcohols and acids by some microbes. Okay. This PDF will be placed to you, so there is no worry in noting down each point. Okay. Enzyme production. Enzyme production, we have many number of types of enzymes and microbes which are involved in producing those enzymes. Like Candida species are involved in producing lipases, which are used as detergents and food processing. Bacillus proteases are produced or synthesized, which will be used in leather and detergent industries. Okay. Streptococcus, streptokinase enzyme, which will be used as clot busters in heart attacks. Okay. So this is important actually, streptokinase enzyme from Streptococcus, which is used as clot buster and heart attacks. Next, microbes usage in medicine. What is the medicinal role of this microbes? Microorganisms revolutionized medicine through the discovery of antibiotics and vaccines, wherein antibiotics and compounds that kill or inhibit bacteria, some microbes also help in producing recombinant molecules, okay, recombinant medicines such as human insulin, which is produced from E. coli. This is the best example for recombinant medicine. Okay. Next. So, antibiotics from microbes. What are the sort of antibiotics that are produced by different microbes? First is Penicillium notatum, which is producing the antibiotic penicillin. Okay, and the targeted disease is what? Bacterial infections. Okay. Next is Streptomyces gracius or Streptomyces aureofaciens. This Streptomyces will produce what? Streptomycin. Streptomyces aureofaciens will produce which antibiotic? Tetracycline. All the examples are important for us. So this is working on tuberculosis. This tetracycline has broad spectrum of usage in different diseases. Next, Streptomyces rifamycinia. This is producing what antibiotic? Rifamycin, and this is also working on tuberculosis. Streptomyces erythraeus produces the antibiotic erythromycin, and this is working on the respiratory infections and inflammations. Okay. So this is the examples which are included under the antibiotics from microbes. And then microbes in sewage industry. In sewage industry, we all know both primary and secondary treatment will be there. In secondary treatment, we will be having the usage of the microbes, not in the primary treatment of the sewage. So here in the sewage treatment, which involves the removal of organic waste using microbes, in the secondary treatment we will be using the microbes, aerobic microbes which oxidize the organic matter significantly, which reduces the biological oxygen demand. Okay, this will reduce the BOD, biological oxygen demand in the water bodies, polluted water bodies. So methanogens are used in the final anaerobic sludge digestion, which is useful for the production of biogas. So usually once if we have to recall the sewage treatment, first primary treatment will be there, where we will be removing all the sludges and all, and then we will be having secondary treatment. In the secondary treatment, first aerobic bacteria will be used, where aerobic bacteria will start working out on the organic material, and it will completely digest the organic waste material which is present, which will produce what? Which will reduce the biological oxygen demand in the polluted water body by increasing the aerobic respiration of bacteria. Lastly, anaerobic sludge digesters will be working out. For example, methanogens, and it will be producing what? Biogas. Okay. So here we have to highlight the microbes in the secondary treatment. Next. So primary treatment, of course, no microbes are utilized. Secondary treatment, aerobic bacteria, fungi, sludge, activated sludge is formed. This will decompose organic matter and BOD will be reduced. Okay. And then we have anaerobic digestion like methanogens or methanobacteria. This will convert the sludge into biogas, okay, which is methane rich. And then microbes as biofertilizers. Biofertilizers are living microbes. This will enhance soil fertility by fixing atmospheric nitrogen on mobilizing nutrients like phosphorus. They are eco-friendly alternatives to chemical fertilizers by promoting sustainable agriculture. So to increase soil fertility, to reduce soil erosion, to increase the organic farming types, what are we using nowadays? Biofertilizers we are using instead of pesticides and insecticides. So in biofertilizers, what is the role of microbes and what are the microbial sources? So if we have to begin with, first is rhizobium. Rhizobium leguminosarum. This has symbiotic association with leguminous plants like pea, bean, wherein rhizobium has a symbiotic association, obligate symbiotic association with the roots of legume plants, where it will form root nodules for nitrogen fixation. Okay. So this is symbiotic association with the roots of legumes for nitrogen fixation in the root nodules. Of course, we all know the mechanism over it, but here we have to highlight the examples. Next, Azospirillum and Azotobacter. Here which is free living, which is symbiotic should be in our mind. Azospirillum and Azotobacter, these are again free living, which are usually linked with non-leguminous plants. This main purpose is also nitrogen fixation. And then if we have to see Anabaena and Nostoc, these are cyanobacteria which are blue-green algae. These are also either free living or symbiotic, and their main purpose is nitrogen fixation. This will increase the amount of nitrogen fixation, especially in the paddy fields or rice fields and in water bodies as well, and this will conduct oxygenic photosynthesis only. Next, mycorrhiza or glomules. Mycorrhiza, the name itself is saying this is fungal association with roots of higher plants like pinus, wherein this is a fungal symbiont with tree roots like for example pinus, wherein this absorbs phosphor and water. Like what happens is, for example, let us assume this is a taproot system of pinus, wherein because of this fungal association, this will increase the surface area of the roots. With the increase in the surface area of the roots, it can easily absorb water and minerals. So to increase the level of absorption, this fungal association is helpful. Okay, and then microbes as pesticides. Biopesticides are microbial agents which are used to control pests. They are safe for non-target organisms and they do not leave harmful residues in the environment. The most common example is Bt cotton and fungal biocontrol agent. Microbes like Bacillus thuringiensis, it is well known for everywhere we will see this Bacillus thuringiensis. What type of toxins it will be producing? It produces cry proteins like cry1AC, cry2AC and cry1AB. So this will work out on what? Bollworms, cotton bollworms it will be working on. So this will kill this bollworm type of insects. Cry1AC kills what type of insects? Cry2AB what? We will be looking into MCQs once. Okay. Next, Trichoderma species, this play key role in fungal biocontrol agent. Okay, where this main target is soil borne pathogens, and then is nucleopolyhedrovirus, which is a viral insecticide. This will kill the caterpillars and borers in the crops. Fine. So this is all about microbes which are acting as pesticides. And microbes in biogas production. Biogas is clean renewable fuel which is produced by methanogenic bacteria from organic waste under anaerobic conditions. Okay. This contains methane which burns efficiently and this is widely used in rural households mostly. Microorganism is methanobacterium or methanogen. This will function as anaerobic digester of organic molecule and this is producing biogas. Biogas is mostly used as a fuel, cooking fuel for many villages. And then microbes in environmental management. Microbes help to detoxify pollutants in air, water, soil as a process of the bioremediation. Okay. So this bioremediation concept is new to us, but then here also we have to highlight examples only, definitions and examples. So to detoxify pollutants which are in air, water and soil, that is bioremediation. They can degrade hydrocarbons, heavy metals and other toxic particles also. Some algae and fungi will absorb and immobilize harmful chemicals from the environment. That is the role of the microbes or the organisms which are playing in the bioremediation. They will absorb the harmful chemicals which are immobilized in the environment, but they will be absorbing that, thereby reducing the pollution. Okay. So first application if we have to see, Pseudomonas putida. Okay. What does this clean? This will clean up the oil spills. Function, this break downs the hydrocarbons which are in the oil spills. Second is algae and fungi, heavy metal absorption, okay, wherein this will absorb the immobilized metals. Lastly, thermophilic bacteria and fungi which are used in composting. Composting also reduces pollution and increases the soil fertility. So this degrades the organic waste into a manure. Okay. Next, microbes in biotechnology. Microorganisms are the foundation for rDNA technology, recombinant DNA technology. They serve as host, vector, sources of enzymes and in gene cloning, production of genetically modified organisms and development of vaccines. Okay. So if you have to see microorganisms, Agrobacterium tumefaciens, this will transfer genes to dicots, okay, by what vector? Ti plasmid vector, which is present in the plasmid DNA. And then Escherichia coli, it is used in gene cloning, recombinant insulin production. Lastly, Thermus aquaticus, this is a source of Taq polymerase enzyme which is functional at higher temperatures also in polymerase chain reaction, that is PCR. So all these are the examples. Here just gist and lines of that are given. In our continuous chapters, we are going to exercise this in more detailed way, where in biotechnology chapter we will be learning clearly about gene cloning, how production of genetically modified organisms happen, what is the role of Agrobacterium tumefaciens, Ti plasmids, everything we will be learning. But here in this first chapter, we have to highlight the examples itself. Now, branches of biology and associated biologists. We have many different branches of biology where the simple definition of biology goes with, as this is the Greek term where bios means life and logos means study. Over centuries, biology has evolved into multiple branches. This will address many different aspects of life sciences study. So, let us look into each of them with the eminent biologists once. Botany is study of plants, we know. Father of botany is Theophrastus. Zoology is study of animal, Aristotle, symbol we know. Next, microbiology, it is the study of microbes by Louis Pasteur, who is the father of biology. Cytology is the study of cells, which is given by Robert Hooke, who coined the cell, that is dead cell. Genetics is the study of heredity, we know Gregor John Mendel. Molecular biology, which is molecular basis of inheritance or life. This is given by whom? Watson and Crick, who gave us the DNA structure as double helical model from X-ray diffraction studies. And then embryology, which is the study of embryo development. This is given by Carl Ernst von Baer, who is the father of embryology. And then physiology, which is the study of bodily functions. It is given by Claude Bernard, who is the father of modern physiology. Taxonomy, classification of organisms, we know where well-known scientist, that is Carolus Linnaeus. He is the father of taxonomy. Say. Taxonomy? Taxonomy of angiosperms. It will be produced in the table, I will tell you later, okay. Evolutionary biology, study of species evolution from Charles Darwin, theory of natural selection, in that book he has published. Next, immunology, study of immune responses by Edward Jenner, which is developed smallpox vaccine. Parasitology, study of parasites by Francisco Redi. Pathology, study of diseases by Rudolf Virchow, who is father of cellular pathology and who modified the cell theory. Then mycology is the study of fungi, which is given by P.A. Macaulay. And then study of viruses by Bajering, who discovered tobacco mosaic virus in virology. Next, study of organisms in environment, that is ecology. Ernst Haeckel is the father of that. For biotechnology, Carey Mullis. For anatomy, it is Andrew Vesalius. He is the father of modern anatomy. So, these are the special titles and contributions in the biology that are given to the eminent biologists. Father of biology is Aristotle, medicine is Hippocrates, genetics will be Gregor Mendel, father of modern botany is Linnaeus, father of modern zoology is Cuvier, father of Indian ecology is Ramdev Mishra, father of modern biology is Charles Darwin, father of modern genetics will be T.H. Morgan, plant physiology will be Stephen Hales, Indian father will be J.C. Bose. Father of DNA fingerprinting, Lalji Singh. Father of tissue culture will be Haberlandt, and who is the inventor of microscope, that is Anton van Leeuwenhoek. So, this is the basic list which I have given for eminent biologists. Once again I will give you the revised list, because there are n number of branches again, as you said, for taxonomy, angiosperms, and Indian scientists as well. Including that, I will produce 2 more tables. After looking into that, we will be solving 50 objective from this chapter 1, and then we will be moving to the next chapter, that is diversity of life forms. Okay. So, till here today's session will be done, and tomorrow we will be exercising this chapter, going for diversity of life forms. From there the concept starts actually. Okay. Thank you.